Welcome back, dear friends, to the BIOZEE classes. In today's session, let us discuss about the continuation of the chapter Human Reproduction. Let me remind you, in the previous class, we have completed the fertilization process. And we know what is fertilization. That is, it is an event or it is a process for the fusion of male and female gametes to occur. The sperm and ova, if it has to fuse, all the steps or the event which is occurring is referred to as fertilization, and the instance at which it fuses is referred to as syngamy. So dear friends, this we have studied in the previous classes. In today's class we are going to discuss about one more interesting topic, an important topic called cleavage and embryogenesis. So first is cleavage, followed by one more important topic, that is embryogenesis. So let me tell you, dear friends, that what is actually cleavage. Cleavage can also be referred to as the mitotic divisions. So the zygote, after its formation, will undergo the mitotic divisions or the cleavage to form an embryo. So this we refer it to as embryonic development or embryogenesis. So let me repeat this point. Embryogenesis or embryonic development is the process of formation of embryo from the zygote. So how it has to occur? That it has to occur through cleavages. Before moving to the actual topic, let me tell you, here cleavages are very simple and they are holoblastic. That means to say holoblastic here refers to that, the whole egg divides. Fine, dear friends. Now you see the picture which is being shown on the screen. So now let me tell you that cleavages, how they occur. Initially, let me tell you, first there is a ovum. In fact, so these cleavages start to occur in the oviduct. See, let me show you the diagram here. So oviduct or fallopian tubes you can refer it to as. So after the release of the egg, in fact you can find here the first zygote formation will occur in the between the ampullary isthmic junction B and C. In between that there is the fusion of male and female gametes. That is in the junction of ampullary and isthmus. So further, dear friends, what happens here, after the formation of zygote, the zygote will keep dividing in the fallopian tubes to form, I am representing here on the screen, please just observe there, it forms the 2-celled, then 4-celled, 8-celled, in fact, and 16-celled embryo. Almost all this occurs in the fallopian tubes, dear friends. So the 8-celled stage, the solid mass of blastomere, what you refer it to as 8-celled stage, we call that to as morula. We call that as morula, as you can observe here. So this is ovum. So it later, after the fusion with the sperm, it forms the zygote, then undergoes the mitotic division or cleavage to form the 2-celled stage, then it forms 4-celled stage and 8-celled stage. 8-celled stage here is generally represented as morula. So in fact, let me tell you that still in the morula we can find the zona pellucida layer covering it. So fine, dear friends. So let me tell you that the outer cells of morula are much smaller when compared to the inner cells. Inner cells are much larger. So inner cells you can observe which are very big. The outer cells are much smaller when compared to the inner cells. Dear friends, let me tell you now further that the young embryo we can represent morula as, in fact, after it reaches the uterus, see here, see the diagram. So after it reaches the uterus further, dividing continuously, that is 16 cell stage, and further it moves on. So when it keeps moving, so the corona radiata, which is covering the zona pellucida layer, will dissolve. So here now we can say that the embryo, 32 cell stage minimum or 64 cell stage embryo is ready to get itself attached to the endometrial lining. Here you can observe H here. So we call that as the blastocyst. So dear friends, let me tell you that the endometrial cells are very full of nutrients, and in fact they are the inner lining of the uterus, we all know. The outer layer is perimetrium, the middle layer is myometrium, and the inner layer is endometrium. So let me tell you that the blastocyst gets attached to the endometrial lining of the uterus, dear friends. So now let us understand how the blastocyst actually looks like. The well-developed embryo, we can say rather. So the outermost cells here, dear friends, we can say that too as the trophoblast cells. So in fact, they are very large and in fact flattened cells, and their role is to absorb the nutrients. So the inner mass of cells are called as embryoblast. We call that as embryoblast. Now let us understand that the trophoblast in fact has a fluid filled cavity inside it, that we call it as the blastocell. We call that as blastocell. And the embryo, as I said you, we call that as the blastocyst. So which is minimum 32 or 64 cell stage. Dear friends, let me tell you, after fertilization, that is after the completion of fusion of male and female gametes or the sperm and ova, after 5 days, in fact we can see that the marula becomes, keeps on dividing, and it becomes 3 times much bigger when compared initially when it was developing in fact fallopian tubes. When you compare, after 5 days of fertilization, we can see it becomes much larger, almost 3 times bigger than actually it was before. So further, dear friends, let me tell you that trophoblast, what is the role? The outer layer of the blastocyst, what is the role of it? So it secretes a hormone. This trophoblast secretes a hormone called HCG, that is human chorionic gonadotrophin hormone. So what is the role of this? It is important for implantation. Further we are going to study. That is, implantation here refers to attachment of embryo to the endometrial lining. So it is important for implantation, and the hormone is also important for pregnancy. So these are few important aspects, dear friends, pertaining to the topic, the cleavage and embryogenesis. Remember, so in the coming sessions, still many more topics we need to discuss. Please do like, subscribe and share. Thank you very much, dear friends.